Hello, everyone. I would like to welcome you to today's webinar, Stronger, Tighter, and Faster Designing New Protein Functions. My name is Gemma Garcia. I'm the European Regional Marketing Manager at Twist Bioscience. Now, I want to introduce you to our speaker for today. Olga Khersonsky is Associate Staff Scientist at the Weizmann Institute of Science. Olga did her Bachelor in Science in Chemistry at the Hebrew University in Jerusalem. In 2010, she completed her Master and PhD in Biochemistry at the Weizmann Institute under the guidance of Professor Dan Tawfik, specializing in enzymology, directed evolution, and protein engineering. After a postdoc in the group of Professor David Baker at the University of Washington in Seattle between the 2010 and 2013, she joined the group of Sadel Fleischmann at the Weizmann Institute. Olga's major research interest is to design new and improved functions in proteins. Olga, the stage is yours. Thank you so much.
I'm glad that Twist Company asked me to present my work to you, and I hope that you will enjoy the seminar, and we will have a nice discussion after that. So, as you all probably know, as a very general rule, no natural protein is ever good enough for any biotechnological or biomedical application. Also, there are often very frustrating trade-offs in which, let's say, an enzyme variant that exhibits good thermostability and high expression yields is the one that is less active. So thermostability trades off with specificity or with catalytic rates. And then we'll have to make really difficult choices about which enzyme or which protein we are going to work with. The reason for those problems is that proteins are large and complex molecules, the most complex molecules we know in nature. And because of that complexity, most protein optimization strategies today have relied on the Darwinian or evolutionary paradigm of trial and error. I will just go briefly through the directed evolution. It is a technique for which a Nobel Prize was given a few years ago. It is mimicking the native Darwinian process that can be depicted as getting from the low point, low hill, to the high hill. And a thing to remember is that no decreases in the fitness are allowed throughout the pathway. So in the lab, we mutate the gene and we create a library by mutagenesis, and then we screen or select improved variants of this library. And then all of this is iterative. So it's going all around. And I can tell you from my personal experience is that this process is laborious. And moreover, it's not even always possible because in some systems, you don't have any high throughput screens. So here's a challenge for protein designers. The challenge is how we design new protein functions. Again, the challenge is that the proteins are large and complex, and not just that, they have unstructured regions such as those loops, right? And those are native proteins which don't have much in common with proteins that are designed de novo, which are typically small, and they don't even have those unstructured regions. And if you look inside the protein, it's even more scary because you have all of those interactions and networks, and everything is very dense. So bottom line, real life proteins are large and their sequence conformation space is just enormous. So in our lab, the way we try to solve this problem is that we combine several things. We combine evolutionary information taken from multiple sequence alignment and conformations observed in the native proteins, and we combine them with atomistic calculations made by Rosetta tool. And recently, we've added also machine learning as well. This allowed us to make algorithms that address protein stability and also protein active sites, and I will tell you about both of those methods today. So the first method that I want to start with is PROS. This is algorithm that addresses protein stability. We all know from the textbook paradigm is that the proteins are made in the ribosome and then they just fold to the native state. However, in real life, what is happening is that sometimes most of the protein is misfolded, or at least its large fraction, and then it's aggregated. And this causes low or no expression yields, heat sensitivity, problems with long-term storage, and also immunogenicity and fast blood clearance. So Adi and Sorel in our lab, they came up with the algorithm that tries to solve the stability problem. So the way it works is that at first step, the sequence space is filtered by the multiple sequence alignment. I'm showing here only 3 positions in the protein, but it goes through all the positions in the protein. And then it eliminates identities that are not present in the multiple sequence alignment. At the second step, identities are eliminated based on their energy calculated by Rosetta. And then mutations that passed 2 of those filters, both of them, they're combined to produce several stabilized variants. Here I'm showing the first success story of PROS. This is an enzyme called acetylcholine esterase. It was a very challenging enzyme because it couldn't be expressed in bacteria. And this stabilization process made it expressing in bacteria. So the expression increased almost 2,000-fold. And also the melting temperature increased by 20 degrees. It is important to notice that PROS in most of the cases doesn't affect the function because you can actively avoid mutations in the active sites. So this enzyme was as active as before the stabilization process. Since then, we made a few improvements to the method. So first improvement is that you don't necessarily need a crystal structure now because PROS can be made on AlphaFold models. So Shiran, in her postdoc studies, she did stabilization of fungal enzymes, which are called versatile peroxidases. She managed to express them and also characterize novel fungal enzymes with diverse functional profiles. Also, we extended PROS to deal with membrane proteins, which are often extremely difficult for heterologous expression. So for the first time, we think that the stability principles we learned might extend to all the classes of proteins. And I just want to tell you about several cases that I personally worked on, which are close to my heart. So I hope that will be interesting to you too. We have this method for 6 years now, and those are 2 cases of stabilization or very challenging proteins. So the first protein is called glucocerebrosidase. So even the name is complicated. It was done in collaboration with Tony Futterman's lab. This enzyme doesn't express in mammalian cells even, and now it expresses in a functional manner in E. coli and is going to be applied as a therapeutic enzyme for Gaucher disease. So we made 55 mutations. We obtained 17 degrees increase in the melting temperature, and which was surprising, we also obtained increased activity. Moreover, we took the mutations made by PROS and we called them the good mutations. And we also had like a large list of the pathogenic mutations because this enzyme is involved in the Gaucher disease. So there are multiple mutations which are causing this disease. And those we declared as bad mutations. And then we had a huge list of 300 mutations with unknown effect. So what we did machine learning, and we made a predictor for mutations, which was based on the good mutations made by PROS and bad pathogenic mutations. So you can see here the bad ones and the good ones. And then what is happening is that when you graph this, and you map according to this division, when you map the unknown mutations, you can actually predict which ones are bad and which ones are good. So we tested several mutants of previously unknown effect, and we got the results that mutants which are good, which were predicted to be good, they indeed expressed, and their activity was practically the same as a wild type, while the mutants which were predicted to be detrimental indeed didn't have any activity. So this predictor is working. The second story is my last project. It was done on serum albumin. So bear with me with this long slide. I just want to convince you that this protein is both challenging and interesting. It is the most abundant protein in blood plasma. If you think about it, we have close to 200 grams of this protein in our blood. It has diverse biotechnological applications. It can be a supplement in cell culture, which is used also in the cultivated meat industry. It is used, as you know, probably in molecular biology and all the restrictions and ligation reactions because it prevents adhesion to the surfaces. It is used as a carrier protein for drug delivery and also in cosmetics industry, in blotting, you name it. It is used in the body as a transporter, so it is binding fatty acids and also all the different small molecules you have in the blood, including drugs. It has weak catalytic activities, which can make the drugs active. It has two conformations. One is slightly open when the fatty acids are binding, and one is the apo conformation, which is closed. Currently, most of the albumin is extracted from plasma, but there are problems with that because it is unethical. You don't want to deal with animal product, and B, the preps are not uniform because if the cow has more fat, then it just will be loaded with more fatty acids and so on. There are recombinant sources of albumin from rice and yeast, but we thought that bacterial production would offer advantages in terms of speed and also efficiency, which comes out to cost. But keep in mind that albumin has 17 disulfide bonds, and it couldn't be expressed so far in bacteria. And I will tell you what actually inspired me to start this project. I was on a lecture by Gary Pielak, and he was using BSA for crowding effects. And then he was complaining that he would like to have a more polar patches, and also more, he would like to have positive patches and negative patches. And then I raised my hand, and then I asked, like, so why don't you mutate the albumins that you're using? And then the whole room, they just turned out and looked at me, and I felt like I'm 5 years old. And then he looked at me like, I fell from the sky. And then he told me, but you cannot mutate albumin. You just get it from the cow, and that's how it is. And I couldn't bear with that because in 21st century, you're supposed to be able to mutate a recombinant protein and work on that. And that just inspired me to start the process of albumins. I worked on both the human serum albumin and the bovine serum albumin. I worked on both conformations and took only the mutations that overlapped, so I wouldn't be stuck only with closed conformation or open conformation. And I made several variants with spatially different mutational loads. So the most conserved version had no surface mutations and no mutations in the binding site. And those are mutations which are shown here as cyan spheres. Then variant number 2 in addition to those mutations, it had mutations in the binding sites, which are shown as magenta. This protein with mutations in the binding sites could be just used, let's say, for crowding, because it doesn't have any mutations on the surface, but it has mutations in the active site. So whenever the binding properties of albumin are not important, you could use variant number 2. And variant number 3 could be used, for example, for blotting, because it had all the mutations suggested by PROS, including mutations on the surface. So I will be talking about 3 variants of human serum albumin and bovine serum albumin. And here are the results. We were actually really surprised to see how well it worked. Even variant number 2 with as little as 16 mutations, and we're talking about protein with 600 amino acids, so it's much less than 10, it's less than 10%. It had increased expression in bacteria, and it could be purified to homogeneity. So here you see the lysates. This is a wild type. This is variant number 1, 2, and 3. So you can see that as the number of mutation increases, you can see also increased expression and also increased stability, because here you see the melting points. So the albumins are not, they are quite stable. The melting temperature is 60 degrees, but we managed to increase the melting temperature by more than 20 degrees. And what is nice is that variant number 3, with 73 mutations, its melting temperature was beyond the boiling point of water. So what you could do, here I'm showing, this is the bacterial lysate after boiling. So you could just boil the lysate and then do the quick gel filtration purification, and you got the pure serum albumin. Then we wanted to test the properties of those stabilized variants. So we checked several properties of the designed variants, and we know that native albumins are monomeric, but they also have some dimer portion, because they have a surface cysteine that can cause dimerization. So we tested them, and HSA1 is mostly monomeric, and BSA2 had both monomeric and dimeric states with varying ratios between preps. Then we wanted to test the binding properties of the HSA variant 1, which I remind you didn't have any mutations in the active site. We checked it by ITC, and we tested binding of warfarin and ketoprofen ligands, which are drugs. They also bind to 2 different sites, which are called the solo sites. And you can see that the binding is retained, and in some cases, it's even tighter than in the native variant. Probably it's because the stabilized variant is a bit more pre-organized for binding. So we definitely didn't harm the binding properties. And then we wanted to see whether you could actually use the designed variants in several applications. So first applications that we tested is the molecular biology application. We tested it in restriction reactions with several restriction enzymes, and all the designs are comparable with the commercial albumin. There is no interference with the restriction, and also it doesn't impair DNA integrity, so the proteins are pure enough, and they act exactly as the commercial albumins. And then we wanted to see whether the albumins are compatible with human cell culture. So we tested HEK cells and hybridoma cells. In both of the cases, we didn't observe any toxicity, and overall the designs were comparable to the commercial albumin samples again. To summarize, we obtained albumin designs that solubly express in bacteria, have increased stability, and some of them are even resistant to boiling, and they can be used in cell culture and in molecular biology. So far I spoke about stability, and let's talk about actual function changes. So I will start about enzymes, but as you will see, this method FuncLib is applicable also to binding sites. FuncLib stands for functionally diverse repertoires of enzymes and binders. The work was done with Rosalie Lipsch and Ziva Mizemrel, which are two excellent PhD students. And what FuncLib does, it is diversifying the active sites of enzymes or binding proteins. And what is challenging about those sites is that they're confined and they have, again, dense networks within the tight active site surrounding. So everything is connected to everything. And in such cases, mutations are often dependent on each other, which is called epistasis. So we came up with an algorithm based on the same logic as PROS, but this time it is applied to the active site. And we don't touch the catalytic machinery because it was evolved throughout the enzyme history. And also when enzymes evolve in nature, usually the catalytic machinery is maintained. During design, what we design is the active site residues which don't participate in catalysis, but which are involved in substrate binding. We can work on a specific substrate if you know how it sits in the active site, or you can also work on apostructure and make a set of diverse active sites and then check it with multiple substrates. So our test case was an enzyme which is called phosphotriesterase. It is a hydrolytic enzyme. It hydrolyzes a wide range of substrates. It is a metalloenzyme which has two metals in the active site. They're showing here. So our catalytic residues were the chelating residues, and we didn't change those. Instead, we randomized 8 positions in the active site, which are responsible for binding of the substrate. Overall, we computationally calculated few thousands of designs, but we chose the top 63 for experimental characterizations. And here is what we obtained. We checked almost 20 substrates. And this is a summary, and pay attention that the y-axis is logarithmic. And above the bars, we have the number of the mutations. So you can see that with less than 5 mutations, we could reach few thousand-fold increase in activity with several substrates, which is 3 orders of magnitude. Among those substrates, there are esters, lactones, and also organophosphates, which are nerve gases. And what is even more impressive is that we have now several variants that can work together as a cocktail and can hydrolyze a range of nerve agents. And they can be used for detoxification, which was a long-standing goal for several labs. Also, take a note that with directed evolution, you would A, need many rounds, and B, for every single of the 20 substrates, you would need what you call a separate directed evolution campaign. So 63 variants provided very good results, very impressive results, with a small fraction of an effort which would be required for the directed evolution. And as I told you before, we were expecting epistasis because in the active site, everything is connected. But here we got very drastic epistatic effects. So the bubble size here corresponds to specific activity magnitude with an ester. And in the center, you have the wild type enzyme, and then you have the single point enzymes. And here you have this hero, the H257-W. But then when you combine all of those mutations, in most of the cases, when you combine with this guy, you see decrease in activity. So there is severe negative epistasis. And when you combine all the 4 mutations, you have this guy. So in the directed evolution process, their combination wouldn't be possible. It would be very, very rare event when you would combine those 4 mutations and get an improvement, because when you would add all of those mutations one to another, you would get a decrease in activity, and that's impossible, because then you wouldn't be selecting this enzyme for the other round in the evolution. So we could kind of jump from one hill to another without going through the low point. FuncLib designs can be applied to stabilize protein interfaces. We recently showed the utility of FuncLib on a homotrimeric interface. This is a protein which is called piocyanin demethylase, and it is disrupting bacterial biofilms. So Rosalie made mutations on the interface of the trimers, and the expression of this protein increased. And Shira, she worked on the interface between the light and heavy chain in the antibodies. And Ravit used to design protein-protein interactions in bacterial toxins, colicins. So again, it can be applied to protein interactions and protein interfaces in a various number of folds of the proteins. Since then, we started to collaborate with other labs, and up until now, I made several FuncLib calculations. And now there is PROS web server and also FuncLib web server. And what is amazing is that we start to see publications of other labs who use the web servers, and we didn't even know that they're publishing those papers. And then, oops, you see that they managed to stabilize this and this protein, and they managed to get increased activity with another protein, and this makes us really happy. So the new thing about FuncLib is that what I showed you before, it involved small sets of designs, which is perfect actually for systems with low throughput. But in principle, FuncLib can be used to construct much larger libraries. And for this, we developed a modification which is called high throughput FuncLib. In this process, we proceed in the same way as with FuncLib. We select positions to design, we apply filters of the multiple sequence alignment and the protein energy, and then we model and rank all the multi-point mutants, but then we don't test top rank designs as before. What we do, we train a machine learning algorithm, which is called EpiNet, on the data of this step. And then we choose only mutations which are compatible with each other. And then we make smaller sequence space. And then we actually design a big library based on this set of mutations. So it can build a library of arbitrary size up to millions. And if you have a high throughput system, it can be applied to this. And indeed, Jonathan in our lab, he made those highly functional active site sequence libraries. He applied it to GFP. He randomized 14 positions which are facing the chromophore of GFP. He made a library of 5 million variants. And within this library, he obtained more than 16,000 unique fluorescent designs, which were not just active, but they also had different spectra, which is shown here. And also it had differences in quantum yield. Some of them were brighter than GFP. And they could be functional in various systems for labeling. As I told you, I hope that I showed you the methods could be used for automated protein optimization. I hope that I convinced you that they were general. Both FuncLib and PROS are available as web service to academic users. Here are the addresses. And two last things, if you are not an academic user, few members of past members of Fleischmann Lab have founded a company which is called Scala, in which Sorel is a chief scientist and I'm a scientific consultant. So Scala licensed the Fleischmann Group technologies, and it works with customers on different enzyme engineering needs, including expression, stabilization, and catalytic activity. And of course, all the genes that I was ordering, they were from Twist. Thanks Twist for that. And I want to thank all the members of Fleischmann Lab. And this is the funding information. And I thank you for your attention. And I will be glad to take any questions.
Perfect, great. Thank you so much, Olga. It was really nice. And we have a lot of questions, actually.
Excellent.
Yes. So first question that was actually quite at the beginning. What is the exact name of the process of the machine learning system that you have used to identify good mutations?
So the algorithms that we're using in the end, the machine learning is used in the high throughput screens, and this is called EpiNet. That's the name of the algorithm.
Good, thank you. Then one question, one person is asking, how does the stabilization work for multimeric proteins and that have several domains?
Okay, so in this case, what you typically do, you go through PROS, but then you restrict mutations at the interface because you don't want the monomers to be soluble but not make any multimer. And if you want to stabilize the interface itself, for that you can do FuncLib at the interface, and then you can stabilize also the actual protein-protein interaction at the multimer interfaces. So it's like two-step process, but sometimes only the stabilization of the core is enough.
Yeah. Then we have a couple of more questions about PROS. Can PROS be applied to proteins that fold via propeptide states? And if so, what approach would you recommend for that?
Propeptide is that when they're processed, you mean? What you could do, I think that I understand this. As in case when that would be the case when you have several conformations. So I would suggest making PROS on each one of the conformations and then take the overlapping mutations. So you wouldn't be only stabilizing the propeptide state, but you would be also stabilizing the proteins throughout the processing.
Okay.
I hope it's clear.
Yeah, if not, the person, if could just
rephrase. You could reach me by mail. That's
good. We can share it then. Also about it, is PROS suggesting the most promising mutations in if yes, based on what is this prediction?
So the prediction is made by multiple sequence alignment and then by the energetic filter. So most probable mutations, probably you mean the consensus mutations. So it's not necessarily the most probable mutation. Sometimes the best position actually is not the most probable one, but it is definitely filtered by the filter of the multiple sequence alignment. That means that if you don't see this identity in the protein family at all, it won't be taken. But so sometimes the mutations are back to consensus, but in many cases, the best identity is actually not the consensus one, but still it is present in the multiple sequence alignment.
Okay, so I understand because the same person is asking a little bit later that you can exclude some mutations that maybe you don't want, like for example, those that are on the active sites.
Yes.
Perfect.
Yes.
Good.
You could exclude. You could actually name the positions which are participating in catalysis, which are binding the enzymes. You could also define what is the interacting chain, other chain in a protein, and you wouldn't mutate the interface with that chain. It's very interactive process.
Okay. There is a couple of questions about albumin.
Okay.
One person is asking, how do you compare the price of your recombinant engineered albumin with the natural bovine preparations?
Yeah. So first of all, I think that you should compare our albumin to other recombinant albumins, right? So you don't compare it to the albumin which is taken from cow plasma. You should be comparing our albumin to the albumin produced by yeast and by rice. I don't have an estimate because we are a lab, we're not a production company. But taking into account that everything can be scaled up, I truly think that it is easier to grow bacteria and it's cheaper than to grow fields of rice. It also takes less time.
So actually there's another person that is asking if there is any interest in commercializing your serum albumin variants or the heterologous process.
Yes, we are in touch with several companies about that, and we are welcoming other people to approach us about that. It is patented, of course.
Perfect, good. One question, would FuncLib, sorry if I'm not pronouncing it correctly, work on allosteric sites?
So yes, again, we are here with the question of dynamics. And here again, I would suggest making FuncLib on the two conformations. And if you want to stabilize a certain conformation that is let's say, bound to an allosteric modulator, then you would make FuncLib on that, and you would stabilize that specific conformation. If you want to make FuncLib but maintain the possibility of multiple conformations, then again, you should make it on both conformations and take the overlapping mutations.
Okay.
And then, yes.
Sorry.
Yeah.
Share more? Okay. Because there is another question that might be a little bit related.
Okay.
For the MSA in PROS and FuncLib, is there any advantage to only use protein sequences for a given group of organisms? And then it explains a little bit, it elaborates a little bit. For example, only land plant sequences included in the MSA for an enzyme that has many protein interactions unique to land plants.
Right. So what is happening is that the automatic version, it makes multiple sequence alignment of all the homologous sequences. The closed homologs and, point mutants are excluded, of course. But what you can do if you're interested in a certain subfamily of enzymes, or let's say only from plants or something else, you can upload your own multiple sequence alignment. And then it will work only on your specific multiple sequence alignment, on your specific sequences. And a lot of people do that.
Then we're almost there. There's a couple of more questions. So how do you construct your MSAs, and what sequences do you get included?
So we use BLAST to do that, but it is a bit more sophisticated because we are also taking into account the conformation regions. So lately we improved the multiple sequence alignment because we are treating the loop regions separately, and it increased the quality of the multiple sequence alignments. So it is based on BLAST, of course. And you can fine-tune how many sequences you get. Usually, due to calculation speed and so on, you limit the number of homologous sequences to a few thousands. but you can limit it further. You also can define what percentage of similarity of the proteins you want to include in the multiple sequence alignment. So it's all very modular.
Okay, so I guess that would also cover in some way the question, what would be the protein size limits for the calculations?
So I would say that there is no limit. There was a group, not in our group, but a group made pros on a protein which was more than 1,000 amino acids long. I don't think that, because the sequence space is limited, we're not taking 20 identities for each position, but much less. Then computationally, it is affordable to make the stabilization of even large proteins. So because the sequence conformation space is reduced.
Perfect. So that was our last question. Thank you so much. It's been a great talk. We had a lot of questions, so obviously it's something very interesting for our audience. And just thank you so much for the wonderful talk. It's been really, really good. And thank you everyone for coming, and have a nice day.
Thank you.
Thank you so much.